Hello friends, today we are going to discuss one of the important topic, RAPD PCR. You know, rapid PCR. That stands for random amplified polymorphic DNA. Random amplified polymorphic DNA is one of the variant of PCR. As yesterday's class we have discussed about AFLP, amplified fragment length polymorphism, and today RAPD, random amplified polymorphic DNA. No two individuals, say with respect to your Homo sapiens, have same genomic, that means DNA profile, what we call DNA fingerprint. Each and every individual has their own distinct DNA profile or fingerprinting. Why? Because of polymorphism. That means DNA polymorphism is a crux point for DNA fingerprinting, and it enables us to maintain individual DNA profile. Just like Aadhaar card, we may have our own DNA profile card. And what exactly the polymorphism? Yesterday's class we discussed. DNA fragments exist, let me a stretch of DNA fragment in varying lengths. That means, say I may have my own DNA pattern, X individual may have my own DNA pattern, Y individual may have their own DNA pattern. What are the possible reasons for this varying length of DNA fragments? What exactly the reason for polymorphisms? It's because of, say, SNPs, single nucleotide polymorphism. Now, how they generate? Simply because of mutations. So mutations leads to SNPs, and SNP leads to polymorphism, and they leads to, in this case, they one particular distinct DNA pattern. That means I may have own DNA pattern. That means DNA pattern in each and every human being is unique. Say for instance, what exactly I mean to say? Say SNPs. Say C, G, T, C, A, G, C. This is one individual, and say another individual C, G, A, C, A, C, G. In third position I have T, another guy may have A. That means SNP, single nucleotide polymorphism. Because of just one single nucleotide polymorphism leads to a variety of DNA fragments. They are called polymorphisms. Polymorphic DNA, and because of this we have a variety of genetic variations, and it is a crux point for DNA profiling. And to identify the same, that means to identify the genetic variations based on polymorphic DNA, we have to employ RAPD. That means that this is the object, that means what we call the reason why we have to apply RAPD, because we have to find out this. So that means polymorphisms is the basis for variety, RAPD. Then the most important point, before we go into the topic, we have to understand the technical terms so that we can understand the concept well. There are four different words: random, one, amplified, the second, the polymorphic DNA. We have discussed about these, polymorphic DNA. And what is random amplified? Random amplification means that PCR enabled amplification. What is random? Here, RAPD, that means random amplified PCR fragments, they are the markers. Markers that are DNA fragments of PCR amplified random genomic DNA segments with single type of arbitrary primer. Though it appears to be a complicated sentence, but the whole concept relies on this particular fragment. What exactly I mean to say is, random amplified means, say, take a primer. The entire concept of RLP depends on this single type of primer. Primer, that means is one type of single primer with arbitrary nucleotides. Generally decamer. Before we understand the mechanism, we have to understand the technical terms carefully so that it is easy to understand the concept. Primer, that means the attribution of random. We are using primer, that is a single type of primer. Unlike in PCR, we use two types of primer. This is very important. In PCR, we apply two different kinds of primers, but here that means here forward and reverse. No, here only single type of primer, which are made up of arbitrary nucleotides, and generally decamer, 10 nucleotides, somewhere around 8 to 12. Okay, so typically decamer. Okay, and this single arbitrary nucleotide through PCR amplifies random genomic DNA segments. These arbitrary primers with that means through PCR machinery, it randomly amplified the genomic DNA segments so that we can get the RAPD marker. So I think it is clear. Say the arbitrary primer, arbitrary the word implies. Here we don't know the sequence. This is the beauty of RAPD. Unlike traditional PCR technique, we no need to have any knowledge of the DNA sequence, what we call targeted DNA sequence. We do not have any knowledge about the DNA sequence, and even the primers are also arbitrary. Arbitrary means it can annihilate anywhere. Say whenever there is a possibility of complementarity, it will annihilate. That's it. Anywhere. That exactly correct. Random, and that leads to amplification. Random, anywhere, random amplified because of this arbitrary primer. That leads to RAPD marker. That's important point. So the entire concept relies on this single arbitrary nucleotide that through the PCR machinery amplifies the DNA and random DNA segments. That means random genomic DNA segments that leads to RAPD markers. That means simply the DNA fragments. So this is how the etymology of RAPD occurs. Then regarding the mechanism, generally if you understand this concept, the rest of the concept is same. With respect to particular example, we will discuss. Say take two different DNAs, species 1 and species 2. We would like to establish the genetic variations between these two based on polymorphs. That means based on banding technique, how it ends. Say this is the DNA of species 1 and this of this species 2. Then the primers, I told the single type of primers, say the forward and the reverse primers. This is what exactly the primer, the same here, the same here, and the same here. Same here also, we have to apply. Then the primers, the forward and reverse. Then one DNA fragment will get it. Say for instance, this is a DNA fragment W, this is a DNA fragment X, and this is a DNA fragment Y, and this is the DNA fragment Z of species 1. Say the same case here also. We will get a DNA fragment A, DNA fragment B, but here in case of say when the primer anneals, it leads to amplification. We all knew that. No, with respect to basic PCR technique, denaturation and all that. Thus, I'm not going to the basics of PCR. No? But here, what happens? A mutation occurs. I told SNPs are because of mutations. Say a mutation happens and that leads to, see, here there is no RNA annealing. That means primer annealing. There is no primer annealing. No primer annealing means no amplification. Here these are all amplified products. Amplified. But here no. That means the C segment has no amplification because of mutation. But D have the same. They have a D DNA fragment. So in species 2 we have A, B and D. This one we have W, X, Y, Z. So we got the also somehow we got the say through PCR machine we got the different fragments. Then we have to analyze. This is very important. How we analyze? Say first we have to say take the same to the gel electrophoresis and applying the agarose gel. Run the gel. Say for understanding. Say there are three. This is for W. No, this is X, this is Y, and this is Z. You know, we have discussed the lighter DNA fragments, they move farther towards the end, whereas the heavier DNA fragments, they are somewhere in between. This with respect to species 1 and respect to species 2 is the same. You know, this is A, this is B, there is no C. So here the gradation is depending on the density. The lighter the density, the farther they move. That means since the DNA is a negative charge in a molecule, they move towards the positive. But the thing is, if they are lighter, they move faster. They move further. The same happening. For the understanding, I read like that. Okay. This is for species 2. Here, C, no amplified. That means no amplification. We got, that means there is no similarity between these two DNA samples. Why there is no similarity? Because I told you already, within the species we have distinct DNA profile or DNA fingerprints. My DNA profile is distinct. I mean there is no match with that of another individual. And how about the species? That means the variation. That means there is the dissimilarity is because of a variation. Variation may be because of mutation or polymorphisms. Say SNPs. Because of this, that means I told you already, the polymorphisms is the crux point for identification of genetic variations. So we have to, the entire scenario, what I call the analysis in banding pattern, what you do, you compare these two things. So that means the banding techniques. You say there is no means it is not similar. They are different. Why there is no similarity? Because of variation, maybe because of polymorphism, mutation, or just somehow because of these there is no match. So we establish that this is a distinct. These are distinct. That means distinct DNA fragments, distinct DNAs, and there is no real correlation. How we establish gel electrophoresis, then autoradiography. Then the visualization is samely done by autoradiography. The pattern is same. Run the gel electrophoresis and agarose gel electrophoresis, then subject the same to autoradiography for visualization. Autoradiograms, you know, this is how the issuance. The entire scenario depends on the single primer that is arbitrary, and it arbitrary leads to a random amplification of DNA segments that leads to generation of RAPD markers, and with that we can identify genetic variation. This is the bottom line of RAPD. On paper, that means it appears to it is a flawless technique, but it does have, because RAPD markers are majorly they are dominant markers. They are dominant markers. The dominant marker means we cannot distinguish the homozygosity and heterozygosity. That means no distinguishing of homo and heterozygosity. This is very important. We cannot distinguish. This is one of the bottleneck of RAPD, and though it is inexpensive and fast, comparatively less reproducible. Less, that means result. Sometimes, but less low reproducibility. Low reproducibility. So how we have to overcome this? You know, for everything there will be an alternate solution. We have scar markers. You know, sequence characterized amplified regions. We have, so to overcome these problems, we have scar. The next, you know, hope you understand what I meant to say. To avoid these, that means to overcome these, we have scar. It's like improvised version, you can say. Scar. sequence characterized amplified regions. It specifically cut down the gel bands. This is very important. So that means to overcome the same, we have employed SCAR. And apart from these, there are different variants of PCR and RAPD. There are different variants. A, P, PCR. That means arbitrary primed. Then DNF. DNA fingerprinting. Is another variant of RAPD with AP, PCR and DNF. So with that means if there is an alternate problem, we can employ an alternate solution. Same with that. Just like in a PCR, there are different variants, like the inverse PCR, anchored PCR, race, RAPD, the same here also. Okay. Hope you enjoy the class and you have to learn what the main objective is to serve the poor and needy students. Accordingly, you can learn. Okay. Thanks for watching.